Hello everyone, and welcome to this webinar on analysis of surface-attached biofilms, where we will talk about biofilm testing using the Symcel calScreener Biocalorimeter combined with BioSurface microbial coupons. My name is Per Ledén, Marketing Manager at Symcel and based at the Symcel head office in Stockholm, Sweden. On this webinar, we are privileged to have two presenters with long experience and knowledge in biofilm testing and applications. Starting off will be Stephen Pedersen, biological engineer and VP operations at BioSurface Technologies. He will give you an overview to analysis of surface-attached biofilm and some of the challenges with the traditional methods. Followed by an introduction to the microbial plate assay, including available materials and tips on using these. After that, Fauve Vergauwe, application scientist at Symcel, will give you an overview to calorimetry-based biofilm testing and the workflow used when running microbial testing with a calScreener instrument. She will then also give you examples of data and applications. We will finish off with a Q&A session where you can send in questions. Please note that you might have to be connected with the app for it to work though. The presentation is recorded and the link will be shared after the presentation. With that, I hand over to you, Stephen.
Thank you, Per. Welcome, everyone. Thanks for joining us today. I'm going to go ahead and start off my section, like Per said, and then I'll hand it off to Fauve once I'm done here. My name is Stephen Pedersen. I work at BioSurface Technologies here in Bozeman, Montana. I usually like to start my presentations off with just a quick quote. This one happened to be a nice one because it's from one of the most cited biofilm papers in the world that I could find. It's got over 17,000 citations, so a lot of you are probably familiar with this paper. It's by Kosterson, Stewart, and Greenberg in 1999. The quote is, "Formation of these sessile communities and their inherent resistance to antimicrobial agents are at the root of many persistent and chronic bacterial infections." And that'll play into what I'm going to talk about here in a couple slides. Quick about me, again, my name is Stephen. I graduated from Montana State here in Bozeman in 2015 with a bachelor in biological engineering and a minor in microbiology. And the role I landed myself in is kind of a nice overlap between those two. I still live in Bozeman with my wife and 5-year-old son. I've spent about 11 years in biofilms, including 1 year at the Center for Biofilm Engineering, which is at MSU at Montana State. I worked in the Standardized Biofilm Methods Lab under Darla. A lot of you are probably familiar with Darla's work. And then through that work, I became familiar with the devices that BioSurface designs and sells, and met my current boss and coworker, and I've been here for over 10 years now. The topics that I'm going to go over real quick are, I'm going to introduce BioSurface Technologies as a company to you. We're going to do a brief overview of biofilms for those that maybe aren't familiar with biofilms. I know most of you probably are. I'm going to introduce the Calgary Biofilm Device, or MBEC, a lot of you probably know it as. We're going to talk about limitations of the PEG design. And then I'm going to introduce the microbial biofilm plate assay, which is our product that I'm here to talk about today, before handing it over to Symcel to talk about their product that aligns with it. We're going to talk about the microbial setup and operation, some of the coupon materials we currently offer, why material selection is important and why it's important to have that ability to use coupon materials. And then we're going to talk about, I'm going to hand off my presentation over to Fauve. BioSurface Technologies, we are located in Bozeman, Montana. We were founded here and we're still here, going on 32 years, I believe. We were founded in 1994 by father and son duo Robert and Brian Warwood. Brian is still our president and CEO. He took over after Robert passed away late 90s, I believe. We were originally somewhat of a spinoff from the Center for Biofilm Engineering here at MSU, with a licensing deal, licensing the biofilm annual reactor. We still sell this reactor. It's gone through a few changes over the years, but it's still one of our core products that we offer and that still gets used around the world right now. We still work very closely with the CBE. We do a lot of device development and standardized methods development with them, and it's a really good relationship that we value a lot since we're so close to them. We offer 6 biofilm reactor lines now, including the CDC biofilm reactor, the drip flow, the annular reactor, rotating disc, the bio-inline biofilm reactor, which is our modified Robbins device. A lot of you are probably familiar with that. And then the industrial surfaces biofilm reactor, which is our newest reactor. We also offer 4 microscopy flow cell lines. We have a transmission, a coupon evaluation, a capillary flow cell, and the treatment imaging flow cell. And then our newest product is what we're going to be talking about today, which is our biofilm assay system. At BioSurface, we try to develop all of our products with a few goals in mind. We design, develop, and implement innovative tools for biofilm investigations while maintaining high levels of repeatability and reproducibility. Real quick overview on biofilms. I know a lot of you are familiar with it already. Generally, biofilms are bacteria or other microbe attached to a surface using what we call EPS, or extracellular polymeric substance. It's basically the slime excreted by the microbe. The biofilm can be very difficult to remove, very difficult to kill. We find it almost everywhere on Earth, and there are studies going on studying biofilm grown on the space station right now as well, and how to prevent that biofilm from interrupting space missions, which is obviously pretty important right now. Just a couple of stats to kind of highlight the importance of why we're talking about this research. Current estimates show 1.7 million annual hospital-acquired infections in the US alone due to biofilms. And globally, an annual estimate of $500 billion, $500 billion of damage due to microbially influenced corrosion. And that's going to be corrosion from biofilm inside drinking water pipelines, inside wastewater, things like that, infrastructure. And because of those, antimicrobial and biocide testing needs to be fast and efficient and obviously reliable so that we can try and tackle some of those issues. Introduction to the Calgary biofilm device. This is kind of the standard biofilm assay right now for antimicrobials testing. A lot of you probably know it as the MBEC, minimum biofilm eradication concentration. It was developed in 1999. It consists of a 96 well plate and a lid that fits on that plate. The lid that you can see here on the left here, it's flipped over, has the 96 pegs. Those pegs are suspended in the 96 wells, and then the wells are filled with a growth media. Biofilm grows on the peg, and then you can do your treatment and removal steps. Currently, this device is only offered in a polystyrene. It comes sterilized, but it's not autoclavable because of the material, and so it's a single-use product. The company that manufactures these, Novotech, does offer a few coating options on the pegs that you can purchase it with, including cellulose, hydroxyapatite, and titanium dioxide. The sampling method for the Calgary biofilm device is ASTM E2799 MBEC assay. Generally, the biofilm is grown on the pegs while they're suspended in the growth media. The lid with the pegs on it is then moved over to an antimicrobial or biocide test to test effectiveness. It is then rinsed with a neutralizing agent to halt the antimicrobial activity. It goes through a 30-minute sonication step. The biofilm is then in a suspended fluid. It's diluted and plated for incubation, and then you get a CFU enumeration at the end of that. This table is just to kind of visualize some of the numbers involved here. A full 96 peg run takes 768 dilution wells, or 8 to 10 96 well plates, depending on how you're doing it. It will take about 32 auger plates, doing drip plating for enumeration. 768 pipette tips, or 8 full boxes of pipette tips that have to be refilled and go through all the autoclaving steps, all of that. There's the 30-minute sonication step we talked about, a 2 to 3 day incubation time and an incubator in the lab. And after all of that, your biofilm is no longer intact. It's been removed and plated, and it's gone. Some limitations of the peg design. I've touched on a couple of these already. The surface area of the pegs is about 109 square millimeters per peg, and we'll talk about a comparison to that number here in a second. The material options available, again, the polystyrene is kind of the main material available, and there's a couple of coatings, but that's about it, and they're all single use. The geometry is not conducive for microscopy. It's that rounded peg cone shape, very difficult to put under a microscope. And you end up having to generally break off that peg from the lid to be able to do that in the first place. So it's destructive. It's hard to maintain that biofilm structure while you're doing that. And that's not ideal for microscopy. You can see in this image, some of those pegs have been broken off here. And again, it's a single-use device. So introducing the microbial biofilm plate assay. This is BioSurface Technologies device, kind of our answer to the biofilm assay. Just in comparison with some of those downsides of on the last slide, the surface area per coupon for this is 150 square millimeters per coupon, so about 40% more surface area. It is fewer coupons, it's 48 coupons versus the 96 pegs, but it is a higher surface area and a higher amount of biofilm per sample, per coupon. Our setup here allows almost any material to be used because it's a coupon-based assay here. The coupons are held in with a set screw. Those can be replaced by almost any material that can be found in this sheet style. So right now we offer lots of metals, plastics, but almost any material can be used. The coupons are easily removed, which makes them very easily placed under a microscope for microscopy. You have that flat surface of the coupon. It's much easier to get images from your biofilm growth. And this entire device is made, other than the coupons themselves, the entire device is made out of autoclavable materials. So the whole thing is reusable over and over and over again. The coupon material, a lot of them will also be reusable and autoclavable. It just depends on what the material is that you're studying. Real quick setup and operation. Again, this is a 48 coupon device. The coupons are a rectangular shape. The standard is 0.8 inches long by a 1/4 inch wide. And we have a specialty size I'll talk about here in a slide or two that is specially designed to fit with the Symcel device. It's a 1/2 inch long by a 1/4 inch wide. And then the thickness on these is about 1 millimeter or so. The entire plate, the coupon holder plate, comes with this alignment plate that you see down here. The alignment plate has these standoffs already installed. So this plate rests on the alignment plate down below, and that height that it ships with is perfectly aligned. So when you're setting up all the coupons and you tighten the set screws, you lift that coupon plate up, place it in your 48 well microtiter plate, and those coupons are perfectly aligned inside the wells for your experiment to begin. The sampling methods for these can include either a single tube method, if you're familiar with that. That's kind of the standard for our CDC reactor and our drip flow reactor, things like that. Or you can use the MBEC method for the micro dilution and plating that way. Coupon materials is very important with this. This is one of the huge benefits to this device. it allows you to grow the biofilm on just about any surface that you want to. Currently, we offer coupons in stainless steels, both 304 and 316. 304 is generally a food processing equipment material. You'll see brewery fermentation tanks will be a 304 stainless steel. 316 is generally medical equipment, things like that. We have a carbon steel right now with the hope of it being used for MIC studies, for microbially induced corrosion studies. We have soft rubbers, silicone, Viton, polyurethane. We can also do natural rubbers, things like that. One benefit of this being these coupons being suspended from the top of the set screw is those flexible materials still work great, since they just hang down. They don't need to hold their own weight. And so those soft rubbers work great in this setup. We have some plastics right now, polycarbonate, polystyrene, HDPE, PTFE, which is Teflon. We do have a titanium available in TI6AL, which I believe is a grade 23 titanium, commonly used for implants and prosthetics. We have a wood available, which is super cool. Most of our reactors, we don't have an easy way to make a wood coupon, but in this format, we were able to create a wood coupon, which is available right now on our website. And then, of course, we have a glass, which is a pretty standard surface for biofilm research and allows for bright field microscopy as well. All the rest you can use, fluorescence, confocal microscopy, things like that. But the glass does allow for bright field. Future additions that we plan to introduce as well. We'd like to do a copper and some other metals. We'd like to do a polished, probably stainless steel surface like the 316 to get closer to some of those polished medical equipment surfaces that a lot of people are trying to recreate. We are trying to get a hydroxyapatite coupon available. I know that that's a very commonly requested material used for oral biofilms, for dental biofilms. And then, of course, we're always happy to work with custom materials that our customers come with. Some biomaterials, surface treatments, coatings, things like that. We're happy to work out how to get these coupons made up with them. Material selection, we think is obviously very important and a huge aspect of why this system is great right now. And a couple of reasons for that, just some examples of why you might need to use specific materials versus the polystyrene off-the-shelf MBEC include porous materials like wood, hydroxyapatite, cement. Those pockets inside that porous material might harbor some cells during your treatment that provide opportunity for regrowth of the biofilm after your treatment. And that's an effect that you're not going to see expressed very well when it's just that polystyrene surface versus one of the materials that your product is actually being used on, like a wood surface, right, or a cement surface. Similarly, there might be some pitting, some corrosion effects on some of these metal surfaces, like the carbon steel that we talked about, and that pitting over time can also provide locations for that biofilm to be protected and to stay alive and provide regrowth opportunity. You're not going to see that on that polystyrene. One thing that I think is really interesting with this is, since the carbon steel coupons are reusable, you can see that change over time, experiment to experiment, using the same coupons. You can see that corrosion effect take place and see how that affects your antimicrobial or your treatment. This image that I have here, the A and C here on the left, all of these are from a carbon steel CDC biofilm reactor coupon, actually. But the A and C image are from abiotic experiments, so no biofilm is introduced. That's just the standard corrosion that happens during the experiment. And then the B and D on the right were biotic experiments, included the biofilm, and you can see the difference between the left and the right images in the amount of corrosion. And so you can imagine that will have effect on how your treatment is working on the biofilm, especially over time, from experiment to experiment. There also may be some leaching effects, whether pro or anti-biofilm. that could affect how your treatment affects the biofilm, leaching from the underlying material. When all you're studying is the polystyrene, you're not going to see different leaching effects, obviously, from material to material. I think that's a pretty important thing, even to just be aware of. If that's not your baseline experiment, it's still important to be aware of any of those effects that might be taking place. Getting ready to hand off to Fauve here at the end. Just a quick recap: the MBPA that we make offers robust, repeatable growth on almost any material. The coupon format is very easy to remove, and we have designed it to perfectly fit this titanium vial from Symcel's calScreener. So our half-inch long by quarter-inch wide coupon was specifically designed to fit this calScreener. The product number is this right here, the MBPA 5025, to get that coupon size. We also offer the MBPA alignment plate in the height that will allow for perfect alignment for those. Running this with the calScreener is great because you have 48 samples per MBPA plate, and the calScreener has 32 spots available. So you end up with about 50% extra samples in case there's some disturbance of the biofilm while you're manipulating the samples over. If you drop a sample, you have some bonuses. It also allows for moving some of those over to microscopy or to other sampling while the calScreener is running, and you can get some side-by-side comparison that way from the same experiment, which is really awesome. I just wanted to end it on this comparison table, bringing it back from earlier in my slides, the MBEC versus now the MBPA and calScreen combination here. Again, samples per run, the MBEC offers up to 96 pegs, where the MBPA has 48 samples. The MBEC uses 768 dilution wells, which is all manually diluted. The MBPA and calScreener combo requires no dilution wells. The MBEC requires 32 agar plates that are all drop plated and incubated. The MBPA and calScreener combo requires 0 agar plates. The MBEC requires 768 pipette tips. The MBPA calScreener requires no pipette tips. The time to result is 2 to 3 days of incubation for the MBEC and only a couple hours for the calScreener to run. At the end of the calScreener running, you get your data, and your biofilm is still intact on the coupon. If you'd like to remove it and place it under a microscope still, you can still do that. The biofilm is still there. You can continue an experiment of some kind if you'd like to. The biofilm still exists on the coupon, which is really awesome. But the MBEC, the biofilm is completely removed and destroyed during sampling. Again, coupon material is huge. Polystyrene is the only available material other than a couple of coatings for the MBEC. And for our system, you can do almost any material. And finally, with the calScreener, you are measuring the metabolic activity directly, which you are not doing with the MBEC. Just wanted to finish up here with some citations that were throughout my presentation. You can take a screenshot or you can look at the recording again later. Just want to make sure that was in there. And with that, I will hand it off to Fauve. I just wanted to say thank you to Per and Casper at Symcel for inviting us to come and talk to you guys today. Some of my contact information is right here. If you'd like to reach out to me directly, my LinkedIn and my email are there. You can take a screenshot or find it again in the recording. You can also reach out to anyone at Symcel, and I'm sure they'd be happy to get you in contact with us. Our website is down here at the bottom, biofilms.biz. Thank you for listening, and with that, I'll hand it off to Fauve.
Thank you, Stephen, for the great overview on the microbial system. So my name is Fauve Vergauwe, and as mentioned before, I'm a field application scientist at Symcel, located in Stockholm. I moved here last year to join Symcel after finishing my PhD at Ghent University in Belgium, where I actually worked on biofilms as well, but more in the context of cystic fibrosis. So I'm very happy that I get to continue working on biofilms right now. And I'm also excited to now build further on Stephen's presentation by sharing how we implemented his system and combined it with microcalorimetry to analyze surface-attached biofilms. Okay, so just wanted to give a brief introduction first on Symcel and also on the calScreener and how biocalorimetry works. Then I will go over and explain what the output looks like of our data, its thermograms, and how you can interpret them. And then I will go over the workflow, how we implemented the BioSurface microbial coupons in our system. And I will explain a bit what different types of applications this system can be used for. So first, let me introduce our company Symcel. We are headquartered at Karolinska Institute in Stockholm, but we also have an office in Boston in the USA. And our science development lead, Casper Craig, who many of you might know, he's located in Copenhagen in Denmark. So Symcel actually brings together over 50 experts spanning 60 nationalities, and we're proud to say that we're the global leaders in biocalorimetry, offering diverse isothermal microcalorimetry-based assays for microbial testing applications. To date, more than 50 peer-reviewed publications have used our calScreener system, and actually many of those focus specifically on biofilm research. So this is our calScreener system. It enables us to measure metabolic activity in virtually any sample type. So we use this 48 well plate with titanium vials, and inside of these vials we have plastic inserts that fit in there perfectly to measure your sample, which are disposable, and the vials can accommodate a volume between 100 and 600 microliters. But what is biocalorimetry specifically? Well, essentially all living cells will produce heat through their metabolism, and our calorimeter can detect this heat with an extreme sensitivity down to the sub-microwatt levels. And the vials with your sample, they will sit on these heat sink that will transfer the heat down to detectors below that are very sensitive. So the heat that we will measure at that point, it's directly proportional to the metabolic activity of your sample. And this also means that we can actually use this to study intact biofilms directly within the vials without having to disturb them. So because we measure the total heat production, we can even capture the metabolism that's going on deep inside the biofilm. The raw data we will get are thermograms. So let me briefly explain what these are and how you can interpret these. So a thermogram will plot the metabolic activity or the heat flow in microwatt over time. This is a process you can follow in real time, by the way. And unlike traditional growth curves based on optical density, they actually provide much richer information. So for example, here you can see a traditional growth curve of E. coli in LB medium. And when the culture enters stationary phase, the growth curve plateaus, suggesting that there's no further activity happening there. However, when we look at the thermogram, this actually reveals that there's quite some ongoing metabolic activity. In this case, the first peak corresponds to aerobic growth, using all of the nutrients and the oxygen. But once the oxygen runs out here, there's actually a switch in metabolism where E. coli goes over into a fermentation state. So this is just an example to highlight how these thermograms can reveal very dynamic metabolic states that might otherwise be invisible with conventional growth measurements. What we obtain is actually a very reproducible metabolic fingerprint that's unique to each microbial sample. So for example, here we show an E. coli dilution series, and you can see that actually the pattern, so the metabolic fingerprint, it remains consistent, but we just see that it shifts to the right as there are fewer cells in your sample. So basically, having more bacteria in the sample, you will have an earlier signal, and if there's less, it will happen later. So this directly correlates to a time to detection, actually. And you can see that you can still detect low inoculum sizes even within hours. So it's quite a fast technique, much faster than most traditional methods. These metabolic fingerprints, they are also very specific to different species. So here I show 4 different species, and you can really clearly see the difference between all of them. But on top of that, it's also very specific to the culture conditions. So here is just one species, E. coli, but grown in 4 different growth media. And you also see a lot of differences. This is just very interesting because this really reflects how the nutritional environments will really shape your metabolism. Then another powerful feature is that microcalorimetry can also analyze polymicrobial samples. So by first characterizing your single species thermograms, for example here, Staph aureus and Pseudomonas aeruginosa, of which you see they really have like 2 distinct thermograms, we can then identify both of them within a mixed community. So you can really see the 2 different peaks in there. So this hopefully helped you understand the data a little bit, how to interpret it, and what kind of things you can, what kind of information you can get from it. But let me now explain how we can actually integrate this with the microbial system. So the microbial system and the calScreener, they really complement each other perfectly for studying surface-attached biofilms. So as Stephen just explained to you very beautifully, the microbial system provides this really standardized platform to grow biofilm on coupons made of a lot of relevant materials rather than on plastic microtiter plates. And after the biofilms have grown on these coupons, we can couple the assay with the calScreener to then actually quantify the biofilm activity with a very high sensitivity in real time. So we don't have to rely on endpoint assays like plating out and counting CFUs. So this kind of setup also really allows us to compare biofilm formation across different materials. It also allows us to evaluate the effectiveness of certain antimicrobial coatings or test the effect of antimicrobial treatments. So by combining these 2, we actually developed a standardized workflow that is capable of testing up to 33 biofilms in parallel. And this approach will really reduce the hands-on time that you need in the lab because it eliminates this very time-consuming plating steps. And it also minimizes the variability in your work. So yeah, that's always nice. So I will just quickly go over like the really practical workflow, how you can handle this system in the lab. So first of all, you can screw on these coupons into the holder plates, And after that, submerge the coupons into a bacterial suspension, where you will allow the biofilms to form on the surface of the coupons. Then the next day, you can very easily remove the planktonic cells by rinsing and placing the whole plate into a 48-well plate with PBS, for example. And after rinsing, you can place this holder plate on a 48-well plate that contains these plastic inserts. So at that point, you can simply unscrew the coupons and they will land into the inserts. And you can add growth medium or any desired type of treatment into these inserts that you want to test. So once the titanium vials are sealed, you can simply measure the metabolic activity that's going on there in the calScreener. So this assay, it actually has quite broad applications. It ranges from like basic research, has a lot of applications in medical devices, but also a lot of industrial applications. So going into a bit more detail, possible applications might be basic research on biofilm behavior and microbiome research, but you can also use it to study anti-biofilm treatments, including bacteriophage therapies. In material science, it can also assess the biofilm formation on diverse surfaces, and it's also ideal for studying antimicrobial coatings and paints, or for evaluating how well a cleaning procedure worked at removing a biofilm. So it's quite broad. This can also be applied into different fields, so that's quite nice. So now I would like to share some results with you, some data that we obtained when we wanted to investigate how biofilm attachment and treatment response really depends on the material type. So for this, we grew Pseudomonas aeruginosa and Staph aureus biofilms on either stainless steel, brass, or HDPE plastic coupons for 24 hours. And if we look at the Pseudomonas aeruginosa data, we see that it grows really well on stainless steel and HDPE, but it seems to like the brass a little bit less. If we then look at the Staph aureus data, we really see a big difference here. It doesn't like brass at all. It takes hours longer for it to peak. So this really shows that there's an inhibitory effect of the brass, and it's the most pronounced against Staph aureus. Well, the brass contains copper, and that is something that we know has some effect on these bugs. And you can really see it here in the total heat that's produced, that there's a significant difference between those materials. So this is just some data to really show you what the importance is of the material you study the biofilms on. Next, we treated the biofilms to evaluate the response of the treatment. And first off, I will show you the Staph aureus biofilms that were treated with vancomycin. So actually on stainless steel and HDPE, we observe a clear dose-dependent response. So as the concentration of vancomycin increases, we really see a lower metabolic activity. And that's the same here with the HDPE. But then interestingly, if we look at brass, first of all, with the untreated control, we already see a much lower heat there in the untreated sample. So this already indicates that there's an intrinsic antimicrobial activity from the brass. If we then add vancomycin to that, we really see that this enhanced the inhibition across all of the concentrations, and we don't even see active biofilm growth in here. So it's really interesting how the brass had such an impact and how combining it with antibiotic treatment really had an extra effect. If we look at Pseudomonas aeruginosa biofilms that were treated with tobramycin, Then we also see that the tobramycin treatment really had an impact compared to the untreated biofilm on stainless steel. And if we look on brass, we see that there's even a clearer effect here that's more dose dependent. So you can really see that there's a significant biofilm reduction compared to the untreated biofilm. So yeah, it also seems to have a bit of stronger effects on brass than on stainless steel. These results already highlight the potential of this assay across diverse applications. And I already explained to you a bit like what type of applications might be interesting, but one I would like to highlight a bit is one that Stephen also touched upon already, is this microbially induced corrosion. So when the corrosion of materials is accelerated by the microorganism. And as Stephen mentioned, this really has significant costs and a lot of challenges across multiple types of industries. So this is an ongoing project that's still in progress. So actually, we don't have data for this yet, but we kind of developed the framework, and we're very excited to see once we start these experiments how it will look like. But I already wanted to talk about it a bit. So basically, the concept is to leverage the calScreener's ability to detect heat from both the corrosion and your microbial signal from the biofilm, as both of these are exothermal signals. So we plan to pre-corrode coupons for 1 to 2 weeks. These are carbon steel coupons. And then we want to measure 3 different types of conditions. So the first one is either looking at abiotic corrosion by itself. So we would do that by putting a carbon steel coupon in a medium that's rich in saline and sulfate. Then we also want to have a microbial control to just see the microbial metabolism by itself by growing bacteria on an inert surface that does not corrode. And then thirdly, we would like to measure the microbial-induced corrosion by combining these two. So by adding bacteria, growing the biofilm on a carbon steel coupon and adding it in that medium. And then hopefully we can capture the combined heat signal from both the corrosion and the microbial activity. So while we still have to experimentally verify this, we anticipate data that looks a bit like this. So abiotic corrosion here produces like a sustained but gradually declining heat signal. The biofilm alone, we know that this generates quite high metabolic output. And then when combined, we expect to see an even higher heat flow that indicates this accelerated corrosion that's really driven by the microbial activity. So yeah, I'm very eager to start with these experiments soon. This also concludes my presentation. I hope this sparked some ideas for potential applications with this assay with you guys, as I believe the use of it is actually very broad. Yeah, if you'd like to discuss your research or explore just whether certain studies might be feasible, definitely feel free to reach out and contact us. Yeah, and I'm also happy to answer any questions you might have.
Thank you very much, Stephen and Fauve. Yes, now you are welcome to send in questions through the Q&A function. I see that some have come in already. There is a question here relating to the polymicrobial system. How does the biocalorimetry work if their signals overlap in time, deconvulsion? and then how the system differentiates between the polymicrobial systems. So anything more on that, Fauve, to mention?
Well, if you really want to know what signal comes from what, you first need to have the single species thermograms to be able to understand that a bit. But if you have overlapping signals, I think it would also be interesting to really see if you grow them together, do they enhance the signal? Do they have a higher metabolic output, or is it lower? To kind of see these interactions there, if they are synergistic or not. But of course, it's not a method most suitable for identifying what is inside of your polymicrobial sample. There's plenty of other methods that are more suitable for that. But it's really interesting to see the dynamics, how they behave together, and how this influences the metabolism.
Thank you. And there is a question on the seeding ratios of staph and Pseudomonas in that graph. Any more input on that?
The seeding ratio. Well, basically, we prepared a bacterial suspension with I think it was 10 to the 5th CFU per ml, and then we incubated the coupons in about 700 microliters of that.
Okay, thank you. Yeah, you're welcome to send in the questions to me or Fauve later on for more detail on it. Yeah. Let's see. A question to you, maybe then Stephen or you, Fauve. How does this method compare to standard methods and align with these as STM, ASTM, and ISO ones?
Yeah, so the microwell growth and treatment period, so everything before it enters the calScreener, aligns really well with the ASTM method that I talked about that uses the MBEC, which is really nice. And then it just diverges right there at the end. So instead of sampling like you would with the MBEC, removing that biofilm, you would divert and go towards the calScreener instead. But the whole first half of that ASTM method can be adapted to help guide the experiment.
Thank you. And there are questions here on materials, on wood coupons. I think you mentioned this, on wood and on concrete coupons.
So I saw the question on the wood. We can make those right now. We do have the material and we do have it available in the standard size. We don't have a listing for the calScreener size for the wood, but if you send us an email, we can definitely get those made up and sell those right now. For the concrete coupon question, we haven't started working on those yet. We hope to eventually. Likely the way those would work is it would be a one-sided coupon, kind of like our concrete coupons for the CDC reactors. So it would probably end up being like a plastic pocket that we then fill with a concrete on one side. But we're still hoping to get those developed and made soon. Again, send us an email about those and we can try and push development if there are specific requirements for some of those.
Thank you. And I think you mentioned it, but there was a question on if the coupons are reusable after grown on them.
And that's going to depend on the material of coupon being used. A lot of them will be reusable. Most of them will be reusable in some way, but it's more a question of how you're going to sterilize them in between. A lot of the materials like polycarbonates, the HDPE and PTFE and all the stainless steels, those are all going to be autoclavable, so really easy to sterilize and reuse. You'll just clean the biofilm and everything off and then put them through the autoclave to sterilize them. Some of the materials like polystyrene, if you're using a polystyrene coupon, won't be autoclavable, but you can still clean it, remove the biofilm, and sterilize it using something like an ethanol soak, and then reuse it that way if you'd like to still.
Thank you. Here's one. Have you tested hydrophobic surfaces? I'm not sure if it's for you, Stephen, or for Fauve.
Yeah, that might be a question for Fauve.
No, we have personally not tested that. I'm sorry.
A follow-up question there on biocorrosion. Would this be applicable to biomining? It's quite a general one, yeah.
Well, I'm no expert in that field, but yeah, would be very interested to further explore this. I think that might be an option. But yeah, if you're interested, please reach out and we can further have a conversation on this to find out what the feasibility is of that.
And just to kind of add to the end of that, if there's specific materials that we don't already offer, like that are included with the mining that don't necessarily align with the stainless steel or the carbon steel that we already offer, feel free to reach out to us and we can see if we can make a coupon in those specific materials too.
Thank you. I think you touched upon this, but on detection times, and of course this will depend on how many microorganisms there are at start, but what would you say are the typical detection times?
Yeah, so most bacteria can actually be detected just within a few hours, even if you have quite low CFU numbers, as you could also see on the E. coli data. Of course, that's fast-growing organisms, so it always is a bit different with slower growing ones. But we know from other types of applications, like our clinical sterility assays, where C. acnes, a very slow-growing one, is very commonly studied, and there we can detect it in very low numbers under 3 days, which is still significantly faster than with other traditional methods. But when working with biofilm studies, actually, most of the times you already have like a very established microbial community, so the heat production is often quite high from the beginning already. So this means you can observe a signal in as little as 2 hours. But additionally, you can also use it for prolonged monitoring because it really can reveal also like sustained signals or identify like surviving subpopulations that are present in very low numbers, which may become detectable hours later. But yeah, in general, most of the stuff you see within a few hours, and if it's like very, very low numbers, you can definitely see it within 24 or 48 hours. But it's very dependent on the organism, of course.
Is there any work on testing how biofilm growth affects the heat transfer resistance through a material, like its insulation effect? Any thoughts on this?
No, I mean, we haven't tested anything like that. It's an interesting thought, but no, it's something we can take with us and think about.
Quite detailed here. What is the best way to mount the coupons to image the biofilm on it?
Yeah, so I'm certainly not a microscopy expert. Generally, from what I understand from folks who are doing this, is you would just back out the set screw, remove the coupon from the micro weld device, and then you would use, manipulation tool, lay it flat on, an empty petri dish or something, and place it on a microscope that way. So the coupon would just be by itself, laying flat on your stage. I hope that answers that question.
Yeah, and there's a question here on the work detecting heat production without a liquid media, for instance, biofilm on wood. Yeah, I guess it's quite clear that we need this, but Fauve, would you say anything more on it?
Yeah, I haven't tried anything myself without liquid media. I think it might be possible, but the thing is that just like if you have any liquid around it, this really makes sure that it's that the transfer of the heat is a bit more, how do you say? I cannot think of the perfect word, but it's easier to measure. You will have a higher signal, but it might be possible. But also, even if you want to test the biofilm on a wood, maybe you can just put some liquid around it so the transferring happens easier. but no, I don't have any experience with that particularly.
Yes, a question here on complex biofilms. Yeah, I'm interested in multi-species biofilms, for example, grown from saliva samples. Is there any limitation for these complex biofilms?
I don't think so, because we love complex biofilms, especially if it's like, yeah, we also love working with these very complex growth media, because there's no limit to the type of matrix that's in there. So you can measure any type of sample in there. For example, we also have done experiments with some chronic wound medium, or something that replicates the cystic fibrosis context. So a medium that's very, very slimy. So I think with these saliva samples, that definitely sounds possible as well, because yeah, the matrix complexity isn't really a limitation for this.
Thank you. You mentioned it, but then on the workflow again, do the microbial coupons fit the cal vials with the plastic inserts, or only without? Just to confirm again.
They fit them with the plastic inserts, actually. The only thing, if you transfer it to a full plate with inserts, there's like on the inner side of the insert, there's a little rig that allows you to take them with tweezers. And you just want to align that all of these rigs are in the same direction, and then you can fit in perfectly your plate, your coupons inside of the inserts. But that works well.
And then question on can you discuss about the variability between biofilms growing in coupons? Anything more to add on this?
Yeah, I haven't added any data on this. So variability really depends also on what type of biofilm. So I've seen that with Staph aureus, the thermograms were really overlapping super nicely. When I had a Pseudomonas strain that produced a lot of slime, there could be a bit more variability, and that's something that you will have. Of course, the whole variability thing is an issue in the whole biofilm research world by itself. But actually, I think it's quite nice with our system, because it's already very standardized. But if you have a super slimy bug, like one Pseudomonas strain that I tested, and if it has like really strings of slime, then of course it might pull off a bit more or less, depending on that. So there might be some variability, but still you're able to group, if you have, for example, untreated and treated, you will really differentiate the different groups there. So, yeah.
Thank you. Another one here on the materials, biofilm on graphite plate for MFC.
I assume that's questions for me. Graphite is not a material I've had any requests for coupons yet. I'd be happy to look into it. That's not one I've encountered yet, so I'm not sure. But we're definitely happy to look into any material.
See if there is not metabolic activity detected, that means the microbe is dead, or not in all cases. It's a quite general, basic question here.
Yeah, normally if you will provide new nutrients and nothing is happening in there, there should be no bacteria in your sample. Yes, because even with like these dormant populations in biofilms, we can still detect very low levels of heat. So yeah.
Thank you. I think those were the time we had for the questions that have come in. Thank you very much again. And if there are further questions, send them along to me, Fauve, or Stephen, and we are happy to answer them. So again, thanks a lot for joining, and wish you all a good day. Thank you.
Bye.